The broadcast is now starting. All attendees are in listen-only mode.
Hi, everyone. I'm Andy Larea, Senior Manager of Field Business Development at PacBio, covering the Americas. I want to welcome everyone to the PacBio webinar series. Today's topic focuses on how gene editing protocols can benefit from smart sequencing at multiple stages. The plan today is that I'll present a brief update on the new Sequel II instrument and the latest 2.0 chemistry, and then Phillip Tai from the University of Massachusetts will give an update on how he has been using long-read sequencing to better understand AAV diversity. The presentation portion of the webinar will be followed by a question and answer session. You're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days. Please keep an eye out for a follow-up email with a link to the recording. So with that, I'm going to go ahead and get started, and then I'll pass it along to Phil. Feel free to put the questions in either while I'm presenting or while he is, but we'll handle all of the questions at the very end. So I just wanted to give a brief update on smart sequencing and how it is being used and some of its advantages. So really the key advantages, and if you leave with nothing else from my section, knowing that we offer very long reads that are free of systematic errors and where we can achieve very, very high single molecule resolution with very uniform coverage. So those are really the kind of key points. With our new 2.0 chemistry, we're able to generate very, very long reads. So here is some sample data off of a 20 kb size selected library. So that's where the insert size is about 20 kb. And you can see that the average read length, or the N50 for reads, is greater than 190 kb. So we're getting really, really long reads off of these inserts. Now, you may wonder how or what you do with a 200 kb read off of a 20 kb insert. Here's our library process where you take double-stranded DNA, we ligate on these hairpins, and anneal and bind the polymerase on one end. That polymerase will go all the way along that first strand, catch the hairpin, and come back along the other strand. So you'll go Watson and then come back along Crick, and you'll continue to do this until the polymerase falls off. Because we have multiple passes on the same molecule of DNA, we can convert that into very high quality single molecule accuracy. So we call these Hifi reads, and these are greater than 99% accurate. When you look at what this distribution actually looks like, you can see that if you filter to a Q20 or 99% accurate, you actually have a mean accuracy that's above Q30 or 99.9, and that's because these are distributions, and some of these will be even higher accuracy because the polymerase itself went around that molecule more times. So this really opens up kind of a new mode of sequencing, which is historically you had the option of using relatively short reads that were high accuracy, or these are longer, noisier reads. But these new Hifi reads are long. They can be tens of kb in length, but are very high accuracy. So they're accuracy equivalent to Sanger reads, but much, much longer. So how we use these in Amplicon sequencing, you can generate with a single primer set, you can amplify regions up to 10 or 20 kb, whatever it is that you're able to amplify. In contrast, when you're doing this with Sanger sequencing or short reads in general, you'll need to fragment that. So here you only have to carry a single set of primer pairs to be able to amplify the region. Here's an example using this strategy for being able to look at the specific site of a Cas9 modification. So generally, it's thought that Cas9 will act on a site and leave behind little to no scar. In this case, they looked at a larger region, 5 kb, around the gRNA target site, and what they found was there were fairly large rearrangements that could be seen because they were able to use these high quality, longer reads to look at a larger region of the DNA. We have a number of ways to barcode these samples. So whenever you're doing a PCR reaction, we've actually got 3 different strategies, and I would encourage you to check out our website or reach out to your FAS if you're interested in this. One of these is the barcoded universal primers shown on the left there. In this case, you use a locus-specific primer during a first round of PCR that has a universal tag on the back end. During the second round of PCR, you use the universal tag as your priming site, and then that will add on a barcode. We've developed a set of these that use M13 as the specific target to target the region. And so in this case, you're able to look at really very quickly and easily transitioning a Sanger protocol over to PacBio long read sequencing. So this is a poster that was presented at ASHG last year, where we did this in collaboration with LabCorp, and we were able to look at about 400 samples on a single SMRT cell. Work presented more recently at AMP this year has extended that to 800, and they continue to work to increase their ability to multiplex onto a single SMRT cell. The traditional method of doing this is with PCR, given the fact that in the gene editing space, a number of groups are interested in samples that are difficult to amplify. We do have a no-amp protocol that we've been working on, and we have launched this year, that really looks at being able to capture a region using a Cas9 system and enrich for it. So there have already been several publications, including the initial no-amp methods paper that have been published using this process. And you can see that quite often they're looking at repeat expansions and being able to sequence through entire repeat expansions. So we have basically one kit for handling all of these applications. And as I mentioned, we work closely with several of the service providers as well as the FAS team to be able to support any kind of experimental design that you're interested in. So as I mentioned, my section was pretty brief. I just wanted to give an update so that you knew the latest with the 2.0 chemistry. It's now my pleasure to introduce Dr. Phillip Tai, a research instructor at the University of Massachusetts Medical School in Worcester, Massachusetts, who will be presenting along with me today, and he will be talking about more applied uses of the technology in the gene editing space. So we're now switching over to his presentation, and again, please, if you have any questions, type them in and we'll deal with them at the end. Thank you. All yours, Phil.
Okay, can you see my screen?
Not yet.
Yep. Okay, so thank you for the introduction, Andy. And you know, we very much look forward to taking advantage of all the advancements that have been made, you know, by PacBio, because a lot of the work that we are currently using, and the work that I'm going to present, is actually using the old RS2 and SQL system. And so we're very much encouraged of all of the positive changes that come with the SQL2 platform. As Andy said, my name is Phillip Tai. I'm an assistant professor at the University of Massachusetts. I've been working here in the Hore Gene Therapy Center for about 5 years, working under Guangping Gao, who is one of the leaders in the AAV field. And what I wanted to do was to kind of talk about how we've been
Phil,
using SMRT sequencing. Yeah.
Sorry, we still don't see your screen.
Okay. Let's see. Do you see my screen now?
We do not.
If
you go maybe up to the top of your dashboard where you see sharing, and there is a, on the left-hand side, there is a button where you can click on that button to share your screen.
Yeah, I see.
There we go.
So main screen. Okay.
So we're
all right.
yeah.
Sorry about that, guys. Yeah. Yeah. So as I was saying, Yeah, we've been using PacBio sequencing for about 4 years now on several sort of projects that involve the development of CRISPR-Cas9 AAV-based gene therapy applications. And so before I kind of go into how we've been using smart sequencing, what I wanted to do was to give a little bit of introduction to AAV, or adeno-associated virus, for those in the audience that aren't really familiar with these promising vector strategies for gene therapy. So AAV belongs to a dependoparvovirus family of single-stranded DNA viruses. They actually came from a very humble beginning because it was first discovered in the 1960s by Bob Acheson as really a contaminant in adenovirus preparations. It's very small. It's about 20 nanometers in diameter. It's considered to be non-pathogenic with low immunogenicity, and on its own is non-replicating. So it actually depends on the presence of adenovirus to complete its own life cycle. So in the human population, about 30 to 80% of the human population is seropositive for AAV, and this is really a double-edged sword. So what this means is that the current AAVs that have been developed, they are easily transducible into humans because the human population have sort of co-evolved with the class of AAVs that have been discovered. However, the drawback is that if patients have been previously exposed to AAV, this means that they may have pre-existing immunity to the vector strategy. So a lot of what is ongoing in the AAV field is to develop new vector strategies to circumvent this immune response. So what's happening? I cannot, oh, there we go. AAV is once again, a single-stranded DNA virus. It consists of a 4.7 kilobase genome. What defines it really is 2 inverted terminal repeat sequences, which are critical for its replication, as well as it contains packaging signal for inserting the synthesized genome into preformed capsids. So the genome itself consists of 3 main open reading frames. One is rep, which codes 4 overlapping genes, which encode for the replication proteins, again, which is involved with replicating its genome, as well as helicase-driven packaging of the genome into preformed capsid, which is consisted of 3 capsid proteins called VP1, VP2, and VP3. And then a few years ago, a 3rd open reading frame called assembly activating protein was discovered, and this protein actually helps to shuttle the capsid monomers into nucleoli to help to form the capsid. So the central sort of dogma for AAV transduction is really that the AAV capsid is central to its tropism, or its ability to target tissues of interest as well as cell types of interest. They bind to glycosylated cell surface receptors as a primary receptor, along with secondary are different between individual serotypes. They are then internalized through clathrin code mediated endocytosis, and then they can traffic through the cell through endosome vesicles, as well as end up in lysosomes. And during this drastic drop in pH, there's a conformational change in the capsid that allows it to escape late endosomes or lysosomes. And upon its escape, it can then shuttle into the nucleus where it uncoats. So once the single-strand genome is in the nucleus, it can then undergo a rate-limiting step to convert a single-strand genome into a double-stranded genome through second-strand synthesis or strand annealing. However, investigators have also developed these self-complementary AAVs that can bypass the second-strand synthesis step. And once in this double-stranded form, then it can be a template for transcription and translation of therapeutic gene products. What is critical for the persistence of AAV genomes is that they can undergo recombination at the ITRs to form these circular episomal species that can persist in non-dividing cells. And people have shown for a long time that AAVs can actually undergo a very low frequency integration into the host genome, and this is for somewhat a few years been a controversial part of AAV. However, the consensus is that these recombinant AAVs persist as circular episomal species and are indeed safe in gene therapy clinical trials. So in recent years, AAVs have gained a lot of attention based on their overall safety and their efficacy. There are over 140 active and recruiting clinical trials for AAV-based gene therapies. The main tissues of target are the brain, the muscle, the liver, and the eye. And this is owing to the fact that the pre-existing capsids that have been discovered tend to be tropic to these tissues. AAV clinical trials are really predominantly in the phase 1, phase 2 steps. There are some that are in phase 3 clinical trials. And one of the reasons why, these clinical trials are really, really critical is we really need to establish whether these platforms are truly safe and efficacious. And so in recent years, there's been a lot of promise for these gene editing platforms, and the leading sort of platforms is based on these CRISPR-Cas9 methodologies, whereby the expression of a nuclease or Cas proteins that can home into specific regions of the genome by a guide RNA can then undergo double-strand DNA breaks in order to promote gene editing events. So if components of these Cas9 gene editing factors can be packaged into AAV and then delivered into patients, this then becomes a very powerful gene therapy approach, where now you have a fully programmable Cas-mediated editing of DNA or RNA that holds a lot of promise for tackling a range of genetic diseases. And so for the past 5 years, our lab has been intensely focused on developing AAV strategies to deliver Cas9 components for the purposes of curing disease. And we really like to break these down into 4 phases: discovery and engineering. We like to also check that our vector designs are of high quality, so we kind of group these into a category of vector QC. We also like to know that the platform that we've developed has high editing efficiency, and we also like to show that that once cells are indeed corrected, that they are also safe and that there are low off-target events, for example. And a lot of the work that we do is carried on in preclinical non-human or non-human primate studies or in mice. And what I'm proud to sort of say is that a lot of these different aspects of our studies really are central to are dependent on these long-read sequencing technologies. And so what I'd like to do is focus my time and discuss how we've been using PacBio sequencing for our studies related to developing gene therapy tools. So during 2001, really the depth of AAV capsids that were available was really limited to a handful of AAVs. But by 2009, through the work of Guangping Gao under the mentorship of Jim Wilson, he had discovered more than 1,000 individual variants in human tissue as well as non-human primate tissues. This spans 9 serotypes shown here in this sort of cladogram. And a lot of these tend to have cross-species transmission. And really, when we think about what's happened in the past 10 years, we've really seen an explosion of the diversity of capsids that have been developed by our lab as well as by other labs in the field. And really, this can be broken down into 4 primary methods for capsid discovery and engineering. The first is rational design. The best example is grafting of a ligand or some peptide sequence that can bind to a cell-specific or tissue-specific receptor. The second is a method which is really hot in the field now, called directed evolution, where you can use error-prone PCR or a shuffling of pre-existing capsids to develop a high diversity library that can then be screened iteratively in vitro or in vivo. And the last is, or the third is in silico design. These are methodologies that employ bioinformatics or machine learning methods to generate capsids that are not seen in nature. But because I'm a student of Guangping Gao, our lab really does focus on what diversity can tell us about vector biology and in natural settings. So what we've done is you can take tissues from either human donors or non-human primates and probe the diversity of AAV in its natural setting. So really the concept of this is very, very straightforward. Like other viruses, AAV has a somewhat of a degree of mutation, and viruses mutate, of course, to escape the immune system. Furthermore, they may undergo a concept called viral compartmentalization, where they can take up residence in certain tissue types to promote its own fitness. So if we are able to query the presence of AAV variation in individual tissue types, perhaps we can come up with new capsids that can be vectorized for the purposes of gene therapy. So we've been using PacBio sequencing in order to identify these capsid variants in different human tissues from, of course, donors. You can take tissues, perform DNA extraction, and then perform PCR using primers that flank the capsid open reading frame. And what you now have is a very highly diverse, complex library that we can then bioinformatically probe for unique variants that are in these tissues. And so before, these techniques were really laborious because they had depended on traditional methods like Sanger sequencing. And we'd like to say in the lab that these traditional approaches is only looking at the tip of the iceberg, where now SMRT sequencing allows us to get the full variability and diversity of AAVs that can be discovered in nature. And this is, of course, advantageous over other small read sequencing methodologies, because now you bypass the need for sequence reconstruction. So this has been a boon for sort of advancing the discovery of new capsids for gene therapy. But what I'd like to do now is focus on how these AAV vectors can actually be designed, and at the same time, what I'd like to talk about is how we've been using SMRT sequencing to perform quality control on vectors that are produced under different design strategies. So recombinant AAVs are relatively easy to create. You can design these into standard plasmids. So as long as the transgene is about 4.5 kb in size and flanked by ITR sequences, any gene cassette of interest that consists of a transgene and a promoter and other auxiliary factors such as polyadenylation sequences and intronic sequences, these plasmids can then be transfected into packaging cell lines in the presence of other plasmids that can introduce central genes originating from adenovirus that are essential in kickstarting the replication and packaging of AAV. In trans, you can also deliver the essential replication machinery as well as the capsid sequences. And in conjunction with host cell machinery, then you can package vector with your transgene of interest. So when I first came into the field, I was very naive in my expectations for the quality of vector that is produced, because my understanding was anything that was designed to be flanked by ITRs is essentially what is packaged. However, this is not the reality. Because these vectors are produced in cell lines, you can imagine that there is going to be some degree of error. And this has been known for a long time, that you can actually get packaging of contaminating DNAs into vectors, and these, of course, originate predominantly from plasmids that make up, the packaging components, as well as those that serve helper functions. But you can also package genomic DNA coming from the host packaging cell line. So this is sort of the reality, and one of the goals for our lab was really to see whether we can develop a methodology to allow us to screen for these contaminating events, as well as the purity of our vector. And so under sort of collaboration with the team at PacBio, we developed a means of querying AAV vector preparations we call AAV genome population sequencing. We first developed a methodology to screen self-complementary vector, which is essentially a double-stranded molecule that's sealed on one end. You can adapter on the open free end to form a single-strand circular DNA template. We have now developed or advanced this procedure in order to also encompass sequencing of single-strand AAVs. So we know that when producing AAVs, you can package equal ratios of plus and minus strand genomes. These can undergo strand annealing in solution to yield double-stranded molecules with open ends on both sides of the molecule. These can, of course, as Andy had stated earlier, undergo rolling circle amplification to yield reads that can then be subjected to strand-specific consensus reconstruction to then obtain minus and plus strand reads, or contiguous plus and minus strand reads that originate from self-complementary vectors that are sealed on one end. And so utilizing this methodology, we were able to show that indeed vector genomes that are packaged into particles is very heterogeneous. So what I'm showing you here is an IGV display of individual reads that are aligned to a self-complementary vector genome reference here, shown as an example. And so under IGV, reads that are perfect alignments to the reference are in gray, and regions that do not match the reference are colored in single bases. But if you allow soft clipping, what you see is that you can get this sort of a rainbow effect of sequence that are not aligning to the genome of interest. So at the very top of this alignment are those genomes that represent expected self-complementary sequences from wild-type ITR to the mutant ITR, and then the complementary strand. So every molecule that is fully complementary, half of the genome aligns well and the other half doesn't. But when we look at this display, what we can already tell is that there is other populations representing truncated genomes that are also have this self-complementary conformation. So already through this type of investigation, we've used smart sequencing to kind of explain how these truncated genomes arise, as well as start to tabulate its frequency of these truncation events. The other thing that we were able to, of course, identify and characterize are these contaminating genomes. So what I'm showing you here in these histograms is the accumulation of these contaminating reads across the human genome for 3 different preps. And right away we see that there isn't really any enrichment in specific chromosomes that have these contaminating sequences. But what was very puzzling for us initially was that a lot of these genomes that mapped properly to HD38, which is the human genome, we also get reads that are also mapping to the vector genome sequence. Originally, we thought that perhaps there were certain aspects of the sequence in these reads that match both the vector genome sequence as well as the human genome. However, when we started to look at individual reads, we saw a commonality in these sequences. One is that most of the reads, when you took these reads that overlap, that mapped to both the human genome as well as the vector genome, they aligned specifically anchored to the wild type ITR. So what this is telling us is that majority of these sequences have one thing in common, which is that they have ITRs. The second is that we didn't see this phenomenon of these reads sharing sequence similarity between the vector genome as well as the human genome sequence. These, turns out, actually are chimeric genomes. So they arose from some type of recombination event where you have packaging of aspects of the vector DNA that are now contiguous with a human genomic sequence. And so this was validated when we look at individual reads, where, for example, this top read is a sequence that maps to human chromosome 16 sequence, but then on the open end of the molecules, you see very clearly that these share the structural composition of self-complementary genomes, where you have 2 ITRs at the open end. And we even see reads that are a result of 6 recombination events. So these chimeric genomes, although interesting, pose a little bit of a concern, at least in terms of potential safety. One is that we had imagined that these type of packaging events may probably be passive, but knowing that they actually have ITR sequences, or at least about 50% of these sequences have ITR sequences contiguous with the contaminating regions, these serve as a active mechanism for packaging via a rep-mediated packaging. In addition, as I had mentioned before, ITRs are important for the conversion of the linear forms of genomes to the circular episomal sequences. So not only do these are these going to persist in the cell, but if they end up packaging large sequences that encompass either promoter sequences or full open reading frames, then this may pose a safety concern. And although these type of methodologies are able to identify these type of species, know that these really are detected at very low percentages. But on the flip side, they do have some mode of to persist in the host cell once they get into the nucleus. So one of the trends that we saw for these sort of truncation events was that we realized very quickly that they tend to be produced in regions that have high secondary structure. So what I'm showing you here is a study where we looked at the truncation events that were generated by the self-complementary AAV that housed a siRNA cassette to target 5'-luciferase reporter gene. And what we were able to show was that there were truncation hotspots that are really centered on these highly structured regions. In addition, we also saw truncation hotspots that appeared in even the EGFP transgene, which is utilized in a lot of these preclinical studies. And these sequencing tracks, where I'm showing you here, showing a relative read abundance as a function of sequence length, really match with agarose gels. And so this is really showing the accuracy of this type of detection for looking at truncation events. But at the same time, in our lab, we were developing AAV vector strategies to deliver Cas9 components. And a lot of you may be familiar that the guide RNA sequence is really in and of itself defined by secondary structure, these sort of loop sequences that are important for binding onto Cas9 so that the guide RNAs can then recruit these to positions that are target sites for editing. And so to address whether guide RNA sequences or any components of Cas9 can actually cause these truncation events, we subjected 3 guide RNA Cas9 construct designs to the AAV-GP-seq methodology. And what we were able to show is that on their own, the guide RNA sequences don't cause genome truncations. And so that's really the good news, is that AAV strategy coupled with CRISPR-Cas9 editing are fully compatible, and they on their own don't really generate the strong heterogeneity in vector preps. However, this is all dependent on design strategy. So we in our lab, we're also interested in developing dual guide strategies to target multiple regions of the genome to perform, for example, large deletions. And when these constructs are designed in a tail-to-tail orientation to cause in effect a large palindromic sequences, as we expected, this causes a lot of truncation. So this is indicated by this huge peak that we see at around 1.2 kb, which is, of course, an undersized vector genome. But when we design them in a tail-to-head configuration where secondary structure isn't very strong, we do significantly reduce the prevalence of these truncated genomes. Although we do see some species that appear to be truncated, but this is very much reduced when compared to certain designs. So once again, on their own, guide RNA sequences don't cause truncation events, but this is really dependent on the design strategy that is employed. One very interesting aspect of this study actually came from a postdoc that was working on this work. He actually had taken this tail-to-tail oriented construct and then realized that they actually still undergo genome editing events and to create these large deletions. And the reason being is that based on our AAV-GP-seq analysis, we revealed that particles were actually packaging fragments of the vector genome that were functional. So what I'm showing you here are populations that span from the 5' ITR to the first guide RNA sequence, and then vector genomes that span from the 3' ITR all the way to the second guide RNA sequence. So these are actually being packaged as individual units. And so we're sort of glad to see that although by gel and by analysis that these secondary structures do indeed compromise to a degree, we know that these vector platforms still do work, and perhaps we can even leverage these type of packaging events to future generations of vector designs that can actually take advantage of these type of anomalies that we are now just discovering. So what I wanted to do now was to actually touch upon how we've also been using smart sequencing in order to quantify the editing efficiencies of some of our gene editing platforms. So what I'm going to talk about is work that was spearheaded by Dan Wang in the group in collaboration with a few investigators in labs here at UMass. So what he was interested in developing is a Cas9 mediated allelic exchange strategy to repair compound heterozygous recessive mutations. So what compound heterozygous mutations are, it's a class of recessive mutations that are typified by two individual mutation types on the same gene. So just to back up a little bit and to kind of clarify what these class of recessive mutations are, is that recessive mutations, of course, are only yield disease phenotype when you get two copies of mutated genes from each parent. And so it's not always true that the mutation is the same, and therefore it complicates the editing strategy. But if you have individual mutations illustrated here, where one is far away from another, the idea is that if you can create a double-strand break between the two different mutations, you can then promote allelic exchange. And what you then end up is with one gene copy that is fully wild type, while another carries the two mutations. And so because the recessive mutations require that both copies of the genes to be mutated, you effectively will rescue the disease phenotype. So for genes such as FAH, which is a mutation in the tyrosinemia type 1 gene, predominantly seen in the liver, we can easily probe whether this allelic exchange events have resulted from Cas9 mediated gene editing by just standard PCR and by histology. But what if you were reliant on diagnostic readout of editing event of tissues that we know has very low transduction efficiency, such as by AAV, such as the heart. We know also that 2% rescue of wild type levels is enough to meet the therapeutic threshold. And so any sort of small, allelic exchange events is meaningful, especially for the patient, right? And so because standard short fragment read analysis is not conducive to being able to detect allelic exchange, especially from mutations that are separated by 3 exons. The SMRT sequencing strategy, which is of course a long read sequencing approach, is really meaningful for these type of investigations. And so after treatment of CRISPR-Cas9 to target this IDUA gene, which is the gene mutated in Hurler syndrome, we see in mice and in the heart muscle that we can get about a 0.1% mutation-free reads. And you may imagine that this is very, very low. However, it's still enough to reverse the phenotype. And without these type of methodologies developed for long-read sequencing, these type of inquiries for gene editing events are not achievable. In addition, Dan has also been pursuing the ability to create transgenic mice using AAV. And of course, this is a project that's also done in collaboration, actually spearheaded by Jaime Rivera's lab, where you can easily create transgenic animals. So some of you in the audience may know that the creation of transgenic animals is actually very laborious. You need a lot of technical expertise as well as equipment to perform these pronuclear injections. But what we're able to show is that all you need is to isolate early zygotes and then bathe them in AAVs that have packaged CRISPR-Cas9 components. Wait a few days until they're at the stage of morula or blastocyst, and then transplant them back into pseudopregnant females. And so under our platform, which is targeting the tyrosinemia gene, we can then assess the loss of coat color or eye pigmentation to gauge the presence of positive gene editing events. And so as you can see here in these lower panels, we see that there are of course neonates that have lost eye pigmentation, as well as newborn pups that have lost their coat color. And there's some of them that are even mosaic. And this is great in order to get a good visual sort of assessment for positive gene editing. But if you really want to know the frequency of indel events that are occurring, you really need a high throughput sequencing methodology. And so we also were employing SMRT sequencing in order to tabulate the different indel events. And as Andy had suggested, that one of the advantages of the PacBio approach is that you can also detect these large deletions that may not be necessarily detected by other standard means. And so I wanted to also wrap up by saying that we have also been using SMRT sequencing in order to really profile whether these gene editing platforms are also safe, right? So not only do they need to be efficacious, but we also want to know that once patients have received these gene editing treatments, that there are no other adverse effects. So our lab and others in the field have started to discover that when AAV is coupled with Cas9 components, that you do get at very low frequencies specific integration events of the AAV genome into positions that are that double-strand breaks have been generated. So some of you may know that that the Cas9 approach also has some degree of off-target events that are generated. So what I'm showing you here is a chirogram display of integration events that we detected in the mouse liver by LAM PCR and Amplicon sequencing. And what I'm showing you here are these upside down red arrows are positions of integration that we've seen. Here we see an accumulation of integration events that are actually overlapping the region that the Cas9 guide RNA is designed towards. And so this type of methodology not only shows us the ability to pinpoint where the off-target integration events are, but also on-target integration events. And of course, again, this may highlight sort of the limitations of the approach, but we really like to see this as advancing our capability of detecting these events so that we can develop new platforms that have improved safety profiles, right? So those vector strategies that of course limit off-target integration as well as on-target integration. And so with that, I'd like to, of course, thank all of the researchers in the Gao lab and collaborative labs, the Rivera lab, Zamor lab, the Shu lab, who have been involved with our development of our Cas9 AAV gene therapy approaches. Sichuan University for collaborating with us on the capsid discovery projects. The Deep Sequencing Core here at UMass that have done all of our sort of PacBio sequencing. The vector core that produced and made all of our vectors that I described here, and of course the team at PacBio, Michael Ryan, Cheryl Heiner, and Christina Weber for again providing support for this project and answering really a lot of questions concerning, PacBio strategy and AAV, the ability to sequence through AAV genomes. So I think I'm going to turn it back to Andy, and I think both of us will sort of field questions. Thank you very much for your time.
Great, thanks, Phil. We have time for a few minutes, a few questions. Some have come in during the talk. If you have any, feel free to type them in. So one is for the AAV GP-seq, what was the percentage of full-length genomes versus truncated genomes?
Right, so this is a very good question. So we do see a lot of variability prep to prep, so even with the same vector design. So it all depends on, what you've designed into your AAV. So if it basically has very few secondary structures designed into the vector, the percentage of these truncation events are very, very minimal. I would say, less than 10 to 20%. We do see a large degree of these truncation events that are attributed to sequences such as, the poly-A sequence, but those are not very strong factors in generating these truncation events, such as those that I showed here in this presentation, like short hairpin sequences and siRNA cassette designs. And so those are really the ones that really give you the biggest problem.
Another question, could you go into a bit more detail about sequencing single-strand AAV, library prep and analysis? Is it the same method as the self-complementary AAV publication?
So we've actually made a few advances to reading out the single-strand AAVs. So we're right now putting together a manuscript to describe the methodology, and really it's compatible with both self-complementary and single-strand AAV. But effectively, you essentially allow both the plus and minus strand genomes to anneal in solution. With that, you get your open ends of the genome that can then be properly adapted by the Bell adapters.
One other question. So a lot of the work that you did was using the RS2. As it shifts to SQL and then more recently the SQL2 and some of those numbers, do you see that as an opportunity to multiplex, or are you probably going to use that to just increase your sensitivity and your limit of detection?
Yeah, I think both. So since switching to the SQL1, and then our core now has recently purchased a SQL2, so it'll further boost the throughput. But just switching to the SQL1, we were able to multiplex multiple vectors onto one flow cell. So some of the projects that we're involved with now, we test upwards to 14 vectors at a time, and the throughput is good enough to allow us to really profile the distribution of these heterogeneous populations. So I think for standard application, as long as we get about 20 to 30,000 reads per library, that's really where we're shooting for, and that gives us enough representation to really pinpoint, where truncation events are occurring, or the degree of contaminants, or where these chimeric genomes are coming from.
Great. Well, I'd like to thank Dr. Tai for his time today and the presentation. In closing, I wanted to briefly mention 3 things. First, we've recorded this webinar and we'll have it available for download in the next several days, so please keep an eye out for a follow-up email with a link to the recording. Second, our Human Genetics Smart Grant is currently open. If you have an interesting research project that could benefit from smart sequencing, consider applying to win free data, which is always nice. We are accepting submissions through Friday, November 22nd. So for more information, you can go to pacb.com/smartgrant. And finally, I hope you'll join us for our next webinar. You can keep up to date on current offerings by visiting pacb.com/events. So thanks again to everyone, and have a wonderful day. Thank you.
Thank you.